All right, hello, 7th grade. I am Mr. Rogers,
And I'm Mrs. Froman.
and you're watching the video lecture for section 4-3 on more modern genetics now, how we can kind of manipulate DNA, mad scientist style a little bit here. So, and we start off with something you watched the video on last week, which was the way we can get certain traits out of animals. So there's 3 specific ways we can do this. So selective breeding, that was the dogs video, talked a lot about that. Cloning, and then genetic engineering also. So we'll look at these 3 different ways we can kind of get specific traits out of animals that we want. And so our first way is selective breeding. And this is a process of selecting those few organisms with desirable traits for parents of the next generation. So really knowing what the parents have sort of traits and making sure they get passed on from generation to generation. And the big one we looked at was dogs, right?
Yeah, so like when they did those foxes, and they were picking the ones that were more tame.
Tame ones, yep.
And then they ended up changing it.
And all kinds of weird stuff started to happen there, yeah. So we're picking out, looking for those traits, yes.
Then we have inbreeding, and that means you're crossing 2 individuals that have identical or very similar alleles. And we see this a lot in dogs, like the picture there of the 2 golden retrievers. If you want to keep having golden retriever puppies, you got to have 2 golden retriever parents. Otherwise, the puppies are going to be looking different.
Long furry coats tend to be, yeah.
So you're trying to get those specific traits. But then that also increases your chance of genetic disorders because you don't have as many different genes coming into that population.
So you kind of say the gene pool is shallow, more shallow, because we're skimming out some other genes, so we're just kind of narrowing down those traits to a smaller and smaller.
Yeah, because the big dogs usually have problems with hip dysplasia. Certain breeds have heart problems, or a lot of genetic problems.
Okay.
Purebred dogs.
And so another method, besides selecting what type of animals breed together, is cloning. And cloning an organism is genetically identical to the organism from which it was produced. So we can do this with plants, really simple. Like I think the spider plant, and they have kind of the buds that come off.
Yeah.
You just clip off one bud, you put the tail in water, and it'll start to grow a whole new plant. Now the big thing about cloning is DNA is the same. That's kind of the big thing. When something is cloned, the DNA is the same as it was in the original organism. So now with animals, we can clone animals. It's been done. Mice, sheep have been done. Now even pets, I hear, is kind of the new thing.
Clone your dead pet.
Clone your dead pet. So to do this, if it could be done, you take the egg cell out of a host, we would say. It's replaced with the nucleus from the cell of the organism to be cloned. So maybe like some skin cells or things like that. And so that nucleus is implanted into the egg cell into another organism to develop, kind of like the surrogate mother then would grow the fetus of that animal. And so to do this, here's our adult, we'll say pigs. I'll say this pig is really good at producing really good bacon. I'm a bacon fan, if you remember there, yeah. So really good bacon fat, that meat part of the animal there. And here's our female we're going to take the egg from, unfertilized egg. We're going to take out its nucleus. We're going to kind of pull it out. You can see there's no more nucleus. And from the pig with the good bacon, we take some of the skin cells. And we want to take the nucleus out of that cell. And so then here's our egg cell and the nucleus. And we send a pulse through it. And they kind of fuse together. So now this new cell will have this egg cell. It's an egg cell from this cow, but the nucleus of of this pig, and the egg cell from this pig, but the nucleus from this pig.
Right. So there's no genetic relationship between the egg and the pig that's actually carrying the egg, right?
So then, now this new DNA will hopefully grow and divide in the egg cell, and it goes into a surrogate mom to kind of carry it, let it grow and develop. And at the end, we come out with our cloned animal, and this pig will hopefully produce that really good bacon.
Should be exactly the same as that
same
other
DNA.
one.
If we feed it the same, everything, it should be pretty much the same animal genetically.
Lots of bacon.
Lots of good bacon. All right. So that's the overview of how we clone. We'll talk more about that in class specifically there.
Yeah. Then our third way is genetic engineering, which is where you take genes from one organism and transfer them into the DNA of another organism.
Okay.
That can be used. We use it to produce medicines, improve crops. They have resistant to diseases crops, and also cure some human genetic disorders. The human genes, for the medicine part of it, we take human genes and we put them into a bacteria DNA called a plasmid, circular DNA, and that makes that bacteria produce what the human DNA codes for, specifically like insulin. So if you're diabetic, the insulin is actually being manufactured by bacteria.
Okay. And basically, we put that new set of that DNA as a new set of instructions, right?
Right.
So the
It's
instructions
just-
for making insulin.
Yeah, it's just the part that tells it this is how you make insulin, and they put that in the bacteria, and then the bacteria makes insulin,
All right. And you can pull the insulin out, and
and
people can use it.
give it to the people. Yeah. The genes of the bacteria are spliced
Yeah.
into plant DNA to help plants survive cold weather, poor soil, and resist pests.
I've heard
So-
one of this one. There's a new type of rice that rice is supposed to flood sometimes, not all the time. So it's more
Right.
flood resistant. I've seen that before too.
Oh, okay. That's kind of cool. And then we've also spliced human genes into cow DNA, and then they can make the blood clotting proteins that are needed for people that have hemophilia,
Okay.
and it comes out in the milk. And then they take that protein out of the milk and give it to the people with hemophilia.
Wow, all kinds of cool stuff there. So
Yes.
and this looks like a picture of how this. So cytokines. What are cytokines again?
Cytokines is a protein that the immune system needs to be able
Okay.
to communicate to tell it what's good and what's bad in your body.
Okay.
So if you're missing this, you really need to have it. So they take that cytokine gene right there
Mhm.
out of the whole strand of DNA up above it. That
Oh,
big
there's a big
purple
strand,
one.
yep.
Yeah, there we go. They take that little piece of the gene out. They cut that bacteria plasmid, that little round thing, so it's opened up, and they kind of splice it in there.
Okay.
So then you can see that it becomes part of that bacteria plasmid, and it goes into the bacteria, and it starts producing those little cytokines, those little yellow dots there,
Mhm.
cytokines.
Okay.
And then they can extract those out and give them to
Yes.
people for if they need that in their medicines.
Very cool. All right. So what we can do this for also is for gene therapy.
Yeah.
And so we're inserting working copies of a gene into a virus. The viruses are very good at infecting us in our DNA. So, which then gets into cells of people with genetic disorders such as cystic fibrosis, and it tries to correct that disorder, that missing with cystic fibrosis, a couple letters that are missing.
Yeah, and that's again another protein that's not being made
right.
correctly.
So if you get that working gene in there through a virus, it's kind of the delivery mechanism. We can hopefully cure this disease. And we'll look at more of this. I have one using this for cancer research also.
And then there's DNA fingerprinting. And so you think about, you got your fingerprints,
Mm-hmm.
and everybody has different fingerprints.
Yep.
Everybody has DNA that's different. So they can actually take your DNA and cut it into little tiny fragments
Okay.
and use electrical currents that separate those little
Mhm.
fragments, and it makes these little bands, and it creates a pattern, so everybody has a different pattern
Okay.
of their DNA. And this helps in to solve crimes and stuff.
Okay. Very cool. And finally, put this all together for the Human Genome Project. And this is something that's been done. We know where every one of your genes are on all of your chromosomes. You can go and find it, and they can say what gene is on what chromosome and where it's at.
Mm-hmm.
So basically, figured out all those 3 billion DNA pairs. So
Yeah.
all those A, T, C's, and G's, 3 billions of them. 3 billions,
Mhm.
3 billion of them. And it comes out to 30 to 35,000 actual genes that kind of make us who we are.
Yeah.
And this has been done, and now they're doing it for other animals too.
Right.
And this is helping a lot too with a lot of the evolutionary research, like how animals are related,
Mhm.
is matching up genomes from different organisms.
Yeah,
So
so they've actually changed classification of
Yeah,
some animals
based on their
because
DNA.
of their DNA. Yeah.
it's not only our DNA, but now a lot of living things' DNA also.
Yeah.
All right, so we're going to stop there. 7th grade. We thank you for watching. We'll see you along the way.
Bye.
It's a long one.